Welcome to my new video. Today we are going to study about the antigen processing and presentation. Students, it is a very important topic from the exam point of view. It comes in every competitive exam and even in our graduation and post graduation exam. So let's see the antigen processing and its presentation. Antigen processing and presentation is the process of digestion of antigen into smaller peptides fragments and by antigen presenting cells that are then displayed on the surface of the cell via antigen presenting molecules. Like we know MHC class 1 and 2 are the antigen presenting molecule, and for the recognition of lymphocyte, and when the lymphocyte recognize it after the antigen presentation, then our immune system evokes and production of high amount of antibody against that antigen is there. So antigen processing and presentation can occur via three different pathways. There are three pathways for antigen processing and presentation. Let's see. The first pathway is the endogenous pathway or class 1 MHC presentation, and the second is the exogenous pathway or class 2 MHC presentation, and third is the cross presentation pathway. So students, these pathway are specifically presented by every MHC class. Like endogenous is presented by class 1, exogenous is by class 2, and in the third cross presentation both MHC1 and 2 are involved. So we are going to see it one by one. Let's see the first pathway, which is the endogenous pathway, and it is a very complicated topic, students, so I designed it in a flow chart. If we study anything in a flow chart, we will understand it easily, and I also show you the diagrammatic representation, but it is a complex process, so I will try to explain it in a very simple way. So let's see the endogenous pathway. The endogenous pathway of antigen processing and presentation, it utilizes one mechanism, student, which is a intracellular protein degradation mechanism. And in this mechanism, student, the antigen presenting cell degrade the protein and antigen into short peptide by a specified cytosolic proteolytic system called as proteasome. And we all know that this proteasome, what they do, these proteasome break that antigen into small fragments. These proteasome break the antigen into small fragments are made, and these small fragments are made by the proteasome, and it is also called as the immunoproteasome. What they call immunoproteasome. And how these immunoproteasomes are activated? These immunoproteasomes are activated by TN alpha, tumor necrosis factor alpha. Then this TN alpha help to activate the immunoproteasome. The next step of the proteolytic mechanism is the peptide trimming by the aminopeptidase in the ER lumen. So the third step is that the trimming of aminopeptide into endoplasmic reticulum, and we all know that what is the function of the endoplasmic reticulum. So the short fragments are then trimmed into the endoplasmic reticulum. Then in addition to that, the TAP taps in, the TAP, which is also called as the transporter associated with antigen. So this protein or these fragments are transported to ER by TAP, transporter associated with antigen, and this molecule help in the transportation of the peptides. Then in addition to that, this TAP or tapsin system mediate the trimming and loading of the peptide on MHC class 1 molecule, and this tapsin and calcin mediate the loading of peptide on MHC class 1 molecule, and this MHC class 1 molecule will transport it or recycle it. It depend on the enzyme, it depend on the protein which is been fragmented, or we also called as antigen, because we know that the chemical nature of the antigen is that it is mainly proteinic in nature, or a polysaccharide or a nucleic acid. So depending on the affinity of the loaded peptides, MHC class 1 molecule will either be transported to the cell membrane or recycled by a mechanism dependent on the UDP glucose glycoprotein transferase 1. And in this case, a high affinity of class 1 complex, the peptides are transported to the Golgi apparatus to the cell. This MHC class 1 molecule transport the peptide to Golgi apparatus to the cell in order to elicit antigen specific CD8 T cell response. What they do? When the MHC class 1 transport it transport the antigen to the cell membrane, and in order it is done to in order to elicit the response by the CD8 cell, and this CD8 cell then evoke the lymphocyte, and lymphocyte leads to the formation of antibody against that proper specific antigen. Let's see the second one. What is the second one? Second is the exogenous pathway. What is second one? Second one is the exogenous pathway, also called as classical MHC class 2 presentation. So what happened in this pathway? In this case, the exogenous pathway antigen presenting cell internalize the antigen by simple phagocytosis, where the material first bind to the specific surface receptor. And what happened in this pathway, exogenous pathway? The degradation of protein into peptide occur by the endocytic process, and in endogenous pathway it is by the cytosolic enzyme. Cytosolic fragmentation is there, and in this endocytic processing is there. And how the endocytic processing is there? In endocytic processing there have been acidic compartment where hydrolytic enzyme act on the antigen, and then the antigen presenting cell have a unique endosome in MHC class 2, and it help in protein degradation. So the internalized antigen process the different acidic compartment to encountering the low pH in each compartment, and the antigen presenting cell have a unique endosome in the MHC class 2 compartment where the final protein degradation and peptide loading takes place. So but question remains, you are thinking that how the MHC class 2 will do all these things? Yes, so answer for this question is that this APC express both of MHC to its surface. So the answer for this question is, when MHC class 2 molecule are synthesized, the class 2 alpha beta molecule are also synthesized, and this associated with a protein, with a specific protein. Remember student, they are associated with a specific protein called as invariant chain, and this invariant chain, this protein interact with the class 2 peptides, binding to the grooves and preventing the binding of endogenously derived peptide to class 2 molecule. And this invariant chain makes different short fragments and later catalyzed by the clip, and this clip present it with antigenic peptide to plasma membrane, and when these antigenic peptide are present to the plasma membrane, it evoke the immune response by evoking the CD4 cell, and this CD4 cell then leads to the production of specific antibody against this antigen. But the question remains that after binding the peptide, these MHC class 2 molecules, these MHC class 2 form a peptide complex, and it then transport it to the plasma membrane, and where the pH is neutral to make the complex in a stable form, because if the pH will be altered, then the peptide will be not in its proper form, and then the CD4 will not recognize it, and then CD4 will not recognize it, then whole antigen processing process will be not of that benefit. So remember student, the pH should be neutral. The neutral pH of the plasma membrane is there, and then the antigenic peptide complex is presented on the plasma membrane, and CD4 cell recognize it and evoke the lymphocyte to make the specific antibody against that specific antigen. So that's all with the endogenous pathway, and we are going to see the third one, which is the cross presentation. Let's see what is cross presentation. So what happened in the cross presentation? The cross presentation is the process where the antigen presenting cell will divert the antigen obtained by the endocytosis, which is an exogenous pathway, to class MHC1, loading a peptide presentation. So all these three pathways doing a single job of making the antigen process antigen presentation process. So what happened in the cross presentation? The APC, the antigen presenting cell, move the antigen to obtain by the endocytosis to class 1 MHC. So the phenomenon of the cross presentation require the internalized antigen. So remember student, my words, the cross presentation requires internalized antigen, and this internalized antigen is being handled by the exogenous pathway leading to class 2 MHC presentation, and then it again redirected to the MHC class 1. So here I mentioned that both class 1. So here I mentioned that both class 1 and class 2 MHC are involved in the cross presentation. So this cross presentation primarily observed in the dendritic cells, and this is mainly observed in the dendritic cell. And the cross presenting cell contain a special antigen presenting machinery, which is an endocytic cell, which signals both to the MHC. Both MHC have been signaled by the this APC machinery. So two mechanisms are involved here. The first mechanism is that the cross presenting cell contain a special antigen processing machinery that enable the loading of exogenously derived peptide onto class 1 MHC molecule. And the second mechanism is that the specialized endocytosis machinery found on the cell, this machinery found on the cell directly represent the antigen on the antigens of the peptides to class 1 MHC molecule. And the main benefit of the cross presentation is that it captures both viruses and present them to cytotoxic C cell. So how it does? The antigen presenting cell can capture the viruses from the extracellular environment and then process them and activate the cytotoxic T cell lymphocyte that can attack virus infected cell, which inhibit the further spread of the viruses. So students, that's all with the cross presentation. Let's see the diagrams. So in this diagram, it is of the endogenous, and it is exogenous. So as I explained in the flow chart, that in endo MHC1 is involved, and TAP transporter activator of transporter of the antigen is there, which transport this antigen by making the antigenic peptides and it is degraded by the proteasome mechanism and then transferred to the plasma membrane where CD8 cells are activated. And the second exogenous, in which they have the endocytic mechanism where endosomes are involved, and then these endosomes make the peptide complexes, and these peptide complexes attach with the clip. I say earlier in my earlier slide, they are attached with the clip protein, and this clip protein attached with the MHC class 2 and present them to the CD4 T cell, where the evocation of the lymphocytes is there, from where the specific antibodies have been produced. So that's all with my lecture on the antigen presentation and processing. If you have any doubt or difficulty in this, you can ask me in the comment box. I will discuss it in my next lecture. So thank you for watching, and like and subscribe to my channel.